Hello everyone, my name is Wahaj and I've just graduated from Anglia Ruskin University for marine biology with biodiversity and conservation, and today I'll be sharing my dissertation project with you that I carried out in Martha Chagres in Egypt to look at the relationships between the physical structure of the reef, the benthos and what's found on the seafloor, and the biodiversity of two families of herbivorous fish. So let's first look at why I carried out this study. Since the 1900s, we know that on average, around the world, coral reefs have declined by about 30%. One of the main drivers of that is overfishing, since the advent of industrial fishing, and the loss of habitat, especially somewhere like Egypt, where along the whole of the Red Sea coast, there are hotels everywhere and the tourists that come with them. And this graph here on the right shows on top the coral cover between 96 and 2003, and at the bottom the fish species on the reefs as well, and both have been in decline. And you tend to find that when you lose herbivores on a reef, so that's the fish that eat algae, you tend to find that coral cover declines and coral reefs start to be dominated by algae. And algae releases chemicals to inhibit the growth of corals, and it also physically takes up the space that they need, which is especially important for young coral larvae, which then have nowhere to settle to grow new corals. Now, the coral is obviously very important for the reef because that provides all the space for everything else that lives on a reef, hiding places and food for everything else as well. And what you then tend to find is that there is a positive feedback loop. When you lose coral, you tend to lose the fish that live there, and when you lose herbivorous fish that eat the algae, you tend to get fewer corals being able to settle. So it becomes a vicious cycle. So here is Martha Chagres, which is located in the Red Sea in Egypt. And the picture on the right shows that it's a V-shaped reef with the north side of the reef on the top and the south side of the reef on the bottom. And a study in 2015 found that the north side had more parrotfish and the south side had more surgeonfish. And I wanted to find out why. Why it's important is because in the Red Sea there are over 300 species of coral, over 1,200 species of fish, and 10% of these are found nowhere else other than the Red Sea. And because of that, Martha Chagres itself has been protected since 1990 and there's no fishing allowed. Let's have a look at the overall structural framework of my study. So the main big questions that I wanted to answer. The first thing I did was to find out the physical structural complexity of the reef. Then I wanted to find out what you find on the benthos, so the percentage of hard coral to soft coral, sand, things like that. I wanted to find out the biodiversity, the amount of species of different herbivorous fish, as well as the numbers of them, and if that differed between the north and the south side of the reef, and if that changed the deeper you go into the water. And this Venn diagram shows the relationships between each factor that I wanted to find out. I wanted to find out where there are differences and where there are relationships. I'll briefly go through with you one of the methods that I use in the study, and that's rugosity, measuring the surface area of the reef. And traditionally you would do that by laying a 1 meter chain over the habitat. So as you can see in this picture, a chain is being laid on the reef. And as that 1 meter chain drops into the nooks and crannies of the surface, it reduces in overall length. As you can see in the bottom here, a 1 meter chain is reduced in length as it's dropped into the coral structure. And the difference between the two measurements tells you how much surface area there is. But it can be quite imprecise because the chain often isn't fine enough to go into all the small nooks and crannies, and it can be quite damaging where bits of the coral can get snagged onto the chain when the chain's lifted up. So what's the alternative? I wanted to use technology to speed up the process and also reduce the amount of damage done with the survey. So I used 3D modeling. Here you can see a surface of coral and there's a digital chain laid on top of the reef. This method is accurate to about 1 centimeter square, much more accurate than the chain and tape method. There's lower risk of damage because you're not physically snagging anything onto the reef. And here you can see a 4-meter square quadrant where there are 3 chains going down, 3 going across, and that gives you 3 measurements of the surface area. The whole thing takes about 3 minutes to complete, and it was pioneered by Dr. Grace Young at Oxford University in 2017. So here's how I used 3D modeling. First of all, I did interrupted line transects. So along both the North Reef and the South Reef, with 5-meter distances, I laid down 4-meter square quadrats made out of PVC pipes, and I swam over them and filmed them with a GoPro, as you can see in the video here. But the tricky thing is that the camera's direction can only change once while you're filming, even though you have to change several times, because you have to film it in this lawnmower pattern of filming, as you can see on the picture on the right. You have to go over the quadrats several times. And I did this on 55 quadrats on each side of the reef, so 110 in total. So once I had the video, I split each video into about 300 separate images. I then used those images to build a 3D model of that quadrant. And the way it works using this software is to stack the images on top of each other, and where there are overlaps in the images, that's how it builds the 3D model. I then imported that 3D model into another software called Rhinoceros, and the script that Dr. Grace Young developed, I used that to lay digital chains on top of the 3D model to measure different aspects of surface area. And here is what I found. So this is a network diagram giving you an overview of everything that I found in this study. In green, you have represented everything that's found on the benthos, on the seafloor. In blue, you have all the different types of measurements that I took of the physical structure of the reef. Yellow circles represent the 2 families of fish and their biodiversity. The links between the different circles tell you what relationships there are in between different factors. The thickness of the links tells you the extent of the influence they have on each other. And the arrows dictate who influences who. So I won't go through the whole thing with you because there's a lot to go through here. But for example, if you look on the top left, you can see turf and coralline algae in green. There is a link to Acanthridae biodiversity, that's the surgeonfishes, and the arrow is going from the turf and coralline algae to Acanthridae. And this shows that actually it's the presence of turf and coralline algae and the amount of it that drives and dictates how many surgeonfish and their biodiversity is found on the reef. But overall, just at a glance, you can see that the main things that are affecting what's found on the reef is the actual benthos. Those are the circles in green. So whether you have dead coral, hard coral and sand, whatever's found on the seafloor drives all the other species that live on the reef, including the herbivorous fish. So what are the implications of this study? Well, we know everywhere on the planet biodiversity increases with the amount of surface area there is in that habitat. There is always variation in that between habitats and within habitats. But we find that the results that I've found aren't applicable to all coral reefs. And here's why. Here's a picture of a typical coral reef, say in the Indo-Pacific, where close to the surface you have a lot of hard coral that have fine structures creating lots and lots of small nooks and crannies where you would find tons and tons of species of fish there to go with it. But in Marsa Chagres, what I found that the shallow areas were dominated by this coral, Porites nidifera, and these are much more dome-shaped, large corals that have fewer nooks and crannies, and that tends to affect the species of fish that are found there. The other difference was that generally speaking, the deeper you go in a coral reef, you tend to lose that physical complexity of the structure because that's normally driven by shallow corals. But in the Red Sea, in particular, you get these things called coral bommies, which are these tower-like structures of hard coral that are growing towards the surface, and alongside their skeleton that's left behind at the bottom, soft corals, sponges, and other encrusting creatures create a lot of physical complexity, much more than you would find at those sorts of depths in other comparable reefs. So this study shows that there are relationships, of course, between surface area and the biodiversity of the fish, and the deeper you go, and what's found on the sea floor. But it is different from reef to reef and region to region. We need to know that before we are able to change policies that are protecting coral reefs around the world. And that's it. Thank you very much for listening. And if you have any questions, I'll be happy to answer them whenever we have time after this presentation. Thank you.